Chapter 5, Structure and Function of Large Biological Molecules. Organisms are made of organic molecules. Organic molecules are the molecules which are made of carbon, hydrogen, and other elements. So I want to give you a glimpse of what actually we are going to learn in this chapter. Don't think that organic molecules are found only in organic substances and we are purely organic, nothing else. It's not quite true. For example, book, uh, is it organic? Think about it for a minute. If you want, pause and research and on Google and find out whether a book is organic, whether the material that the book is made of, is it organic? Grass is definitely organic. Human, are we organic? 75% of our bodies are water, as water. So, water is not organic. So think about it. This alien ship looking like thing is virus. It's actually a virus which infects E. coli bacteria. So that pink thing that you see is actually E. coli. So virus has DNA. DNA is a genetic material. This outer coat that you see, that is all protein. Bacteria are organic, virus is organic. We are made of organic molecule, but not 100%. So the living organisms, body parts are made of organic molecules, but we also contain inorganic molecules. So the point that I would like to convey here is that cells are made of organic molecules, and these organic molecules are divided into 4 categories. We are going to touch upon the categories little later, but wanted to show you. This is an animal cell, this is a plant cell. Where does exactly the molecule go? These are all molecules. Whatever you see here, the colored parts that they're showing in the drawing, they're all organic molecules. So if you take the cell and magnify it under the electron microscope, you can very well see all these are organic molecules. Okay, except this dark purple part. All are organic. Okay, so our cell membrane is a barrier which is keeping the area of the cell and the contents of the cell inside, and outside contents are outside. So it is acting as a barrier, and inside the cell there's water, outside the cell there is water. So in order to keep the inside contents inside and not letting all the water come in and burst the cell, and the reverse is also true. All the contents going from inside to outside. So the cell membrane must be made of something which will inhibit water. So they are made of lipids. Lipids are those category of organic molecules which do not dissolve in water. So it acts as a good barrier. But then we want some water molecules to pass through whenever we need water, right? And whenever there is excess water, the water should go out as well. Excuse me. So the proteins are acting as little doorways or windows. Our genetic material is made of a substance called DNA. Just like, our clothes are made of cotton or acrylic, like that. Genetic material, our chromosomes are made of DNA. So the 4 classes of organic molecules are carbohydrates, lipids, proteins, and nucleic acids. Each of these organic molecules have the polymer and monomer, except lipids. Lipids do not have polymers and monomers. The polysaccharides are the polymers of carbohydrates. Monosaccharides are the monomers of carbohydrates. Similarly, you can learn for protein and nucleic acid. For each category, you have to know the examples as well. So pause for a little time and make a note of this slide, make notes, and this is a good study material for your test. The test questions could be something like this. What is the monomer of protein? Or give an example of uh, polynucleotide. Hemoglobin is polypeptide or polysaccharide. So questions like that. Get ready for those. Cell is made of organic molecules, and these make the specific parts of cell. So a quick review. Our chromosomes, which carry the genetic information, they're made of organic molecules called nucleic acids, whereas our membrane is made of lipids, and the little exit and entry places or spots or little areas in our cell membrane which will allow charged particles to pass through are made of proteins. So where are carbohydrates? Carbohydrates, they act as fuels, and they give structure to cell by forming the extracellular matrix on outside of the cell. And the blood groups, blood group, whenever you say you're A plus or B positive or O positive, that is actually, whenever you say I'm A or B, that means you're saying that I have a specific kind of tag here. So maybe this is A, and this is B, something like that. So these hangings outside the cell membrane, which are attached to the proteins, they act as markers for your blood group. Okay. So, quick review. There are four classes of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Macromolecules means large molecules. Out of these four classes, only three will form polymers, and other one does not. So let me see this in your notes. Bring in your notes with this point: which ones form polymers, which ones do not form polymers. I would like to give you an analogy for remembering what is polymer and monomer. Think of monomers as Lego pieces. Um, you might have been to Legoland, right? There they make everything out of Lego pieces. So a Lego piece is a monomer. You take many Lego pieces and you make um, a big structure out of it, maybe a skyscraper or a house. Then it is a polymer. You can break down house made of different colors, Lego pieces, and then you can make a dolphin out of it. So polymer is nothing but a big molecule which is made of monomers, and monomer is a small building block or a unit which can be used, with many of which can be used for building polymers. Okay, so coming to the two types of reactions, the dehydration and hydrolysis. As soon as I say hydrolysis, try to remember digestion. And we all know what happens in digestion. We break down large molecules to small molecules, right? So hydrolysis breaks large molecules, which are polymers, into monomers. Dehydration makes large molecules by joining monomers. Some polymers are polysaccharides, polypeptides, polynucleotides. All these are made from their specific monomers. So why are they both containing the word hydra in them? It's because water molecules are involved in these two opposite types of reactions. Look at this, guys. This is dehydration. Whenever your cells are making polynucleotides, that is, new DNA is being made, they're putting nucleotides together. These nucleotides, which are monomers, they are happy as monomers. They do not want to cling together and make a polymer. But then we are in need of making a new DNA, which is a polynucleotide. So, in order to make them clingy or glue to each other, our enzymes, they remove hydrogen from one nucleotide and hydroxyl from another nucleotide, and then these bonds are empty, so they cling to each other. So 3 will join with 4. That is how nucleotides are attached to each other. Remember, hydra is the word which indicates water. So here, look at this. Water molecule is removed, right? That is why this is dehydration. Dehydration makes a polymer. Hydrolysis, as I said, hydrolysis, digestion means breaking means polymer is broken down into monomers, right? Again, this polymer is quite happy, it doesn't want to break down into monomers, but then in your stomach you want to break down large molecules to small molecules. So your enzymes, your stomach enzymes, they hold on to this polymer, and they go between 3 and 4, and they say that I need to chop it up. It will chop it up, but then 3 and 4 want to come back together because they are clingy to each other, right? What enzyme does is it will also make sure that it will keep them separate by adding a H to one side and hydroxyl to another side, or a monomer which is broken. That is how they become separate. And now we can absorb these monomers. Our cells cannot absorb polymers, which are big molecules, but our cells can absorb monomers. So that is the goal of hydrolysis. And this process of hydrolysis does not only take place in digestion, but this takes place inside the cell. Now this molecule can be further broken down into smaller molecules, which can happen inside the cell, and that is required for various processes. So just remember, two types of reactions, dehydration and hydrolysis. Dehydration will, I will let you fill up and bring that in your notes. So what does dehydration do? What does hydrolysis do? And another question, why do they both have the word hydra in it? Otherwise, what is the role of water molecule? Again, what are the two types of reactions? What does each reaction do? And what is the role of water in each reaction? Look at these questions. Look at them for a minute and see if you know every word in this questions, and are you in a position to answer them right now? I bet that you know DNA and protein, but you do not know other words, right? And same thing here. Um, if you know that glucose is a monomer, and they're asking which one is a monomer, what is not a polymer is a monomer, and you do not know much about the other ones, right? So, well, these are the kinds of questions that will be asked. So you have to know each category of organic molecules and the examples of each one, and you should be able to remember all these and connect all these dots in the form of the question in order to answer the question correct. So be prepared for that and study from that point of view. Okay, guys, so let's see how we can answer these questions. Okay, so first category, carbohydrates. What are carbohydrates? What do they do for us? And what all do we know? What all we need to know in carbohydrates? In carbohydrates, we have to know a couple of examples for monosaccharides, disaccharides, and polysaccharides. So carbohydrates, the common name is sugars. The reason why they are called sugars is because most of them are sweet, but not all. Polysaccharides are the polymers of carbohydrates, and these are made of little monomers, and these monomers have a specific name. They are called monosaccharides. What's the purpose of carbohydrates in living organisms? They serve as fuel and building material and many other purposes. You have to remember the formula for glucose, and the monosaccharides, they contain the functional group carbonyl. Try to recollect carbonyl functional group. How does it look like, guys? And here, this is a chart which is showing the uh, all the monosaccharides. As you can see, this beige part, this is something that you should recognize. There are two functional groups here. These are hydroxyls, right? Okay. Well, the good thing is you don't have to remember anything from this chart except glucose. You have to remember the molecular formula of glucose and structural formula. Molecular formula of glucose is C6H12O6, and the structural formula is this picture. Okay, so glucose is linear when it is in when it is dry, whereas it curls up and forms a ring when it is in aqueous solution. When two glucoses undergo dehydration and they form a bond between them, then it is called maltose. Maltose is a disaccharide. Di means two. Saccharides are the sugars. So this linkage between the two monosaccharides, it's a covalent bond, and it is specifically called glycosidic linkage when it is between two monosaccharides. Another disaccharide that you need to remember other than maltose is sucrose. It is made of glucose and fructose. Look at this, guys. This is dehydration again. You have to remember for the test that this is glycosidic linkage. Glycosidic linkage is the covalent bond between the monosaccharides. Polysaccharides, you have to remember four of them. And these four are divided into two categories. That is storage and structural. Storage polysaccharides give energy. Structural polysaccharides give strength and shape to cells. And you have to remember each category of one in plant and one in animal. So learn this slide example for each one, and we will talk about each of them in coming slides. Coming to the storage polysaccharide in plants, example is starch. Plants photosynthesize, they make glucose and store it because they know that during nighttime they won't be able to make food. So they have to be ready. All these extra glucoses that they make through photosynthesis, they are going to polymerize them into starch and store it in organelles called plastids. Chloroplast is a plastid. Just like in cloth, you have cotton, acrylic, chiffon, different material like that. Starch has various forms, and two of them are amylose and amylopectin. Look at this, guys. All these green circles, all shades of green circles, not circles, hexagons, they are all glucoses. They're all glucoses, all of them. As you can see, amylose and amylopectin, they're both starch. They're two different forms of starch made of glucoses. And glycogen is also made of glucoses. If they're all polymers of glucose, then why different names? It's because of the different structure. You see this? They have less branching, more branching. Because of that, they differ. They differ in their properties. Coming to storage polysaccharide in animals, glycogen that we just saw. We make extra glucoses into a polymer called glycogen in our liver and muscle cells, and we store it. Cellulose is a structural polysaccharide found in plants. This is also made of glucose. Hey guys, did you notice that starch is also polymer of glucoses in plants? Cellulose is a polymer of glucose in plants. If they're both polymers of glucose, then why is one for storage purpose and another one serves only for structural purpose? It's because starch is a polymer of alpha glucoses and cellulose is a polymer of beta glucoses. What is this alpha glucose and beta glucose? When this linear structure of glucose is turning into a ring in aqueous solution, then the way it curls up, that makes a difference. The way it curls up, if both hydroxyls face same side, then it is alpha glucose. If hydroxyls of carbon 1 and carbon 2 face opposite sides of the ring, one is above the ring and one is below the ring, then it is called beta glucose. And starch is a polymer of alpha glucoses and cellulose is a polymer of beta glucoses. Remember that? And because of this, they vary. In our stomachs, we have enzymes that will recognize the alpha glucose polymer and are able to break down starch into glucoses. They're able to break down this glycosidic linkages, whereas we do not have enzymes which will recognize the glycosidic linkage between beta glucoses, so we are unable to digest cellulose. So this cellulose um, will simply pass down our gut when we eat, whereas starch we are able to use it because we break it down into glucoses. Look at this, guys. This is a leaf made of many cells, and these cells are under the microscope. They look like this. Each cell has plastids. They store starch, whereas the outside of the cell is covered by cellulose, which is a polymer of glucose. Cells make the cellulose and secrete it to the outside. Cellulose gives strength because of these hydrogen bonds between these linear polymers. They're all together and they give strength to the cell. The cell wall gives nice protection to the cell and strength to the cell, so gives shape to the cell. Plant cells get shape because of the cell wall as well. The citrusel, the fiber supplements that are sold in the stores, pharmaceutical stores, such as Walgreens and CVS, it's actually the fiber. Um, when one is unable to pass stool easily, that means they have to take the citrusel. So actually, they are eating cellulose in these supplements. Anyways, the word cellulite is very similar to cellulose. Don't confuse them. Cellulite is fat. Fat is lipid, whereas cellulose is carbohydrate. Remember 4 classes: carbohydrates, lipids, proteins, and nucleic acids. So cellulose is carbohydrate, whereas cellulite is lipid. They are not same, guys. Remember the use of cellulose. When we eat spinach or vegetables, we are eating both cellulose and starch. We are able to make use of starch. We absorb, we break down starch and we absorb glucoses from it, but we are unable to break down cellulose, so it will just pass down as undigested waste. And when this undigested waste is passing down, cellulose irritates the lining of your intestines, and that causes your intestines to secrete mucus, which is slimy substance. And this slimy substance causes this undigested waste to slip through faster, and that helps in defecation easily. So it helps in smooth movement. Yeah. We are unable to use cellulose, but cows and termites, which can eat paper and wood, respectively, they have bacteria which make enzymes to digest cellulose. So they are able to use it. Humans cannot. Coming to structural polysaccharide, chitin. Chitin is the exoskeleton material, which is actually polysaccharide. This is not lipid, protein, or nucleic acid. The bug's exoskeleton is made of polysaccharide, which is carbohydrate, called chitin. It is made of a derivative of glucose like this. So we take this chitin from exoskeleton of bugs, and we make surgical thread, and the surgical thread can be used to sew up people after surgery. And this surgical thread, it will naturally decompose, and the wound heals. The person need not go to hospital to get the stitches removed or staples removed like in olden days. So that is uh, the development in medical field. We are making use of a polysaccharide in such a way. Coming to the end of the carbohydrates, I would like to um, emphasize on some of the things that you need to learn. So I want you to answer these questions and write it down in your notes and turn it in for extra credit. Next category is lipids. Lipids do not like water because they have non-polar covalent bonds. This is a very important point, guys. This is something that we learned in earlier chapters. There are three types of lipids: fats, phospholipids, and steroids. Fats, example is triglycerides. Steroids, examples estrogen, testosterone, and cholesterol. Try to remember these examples. We have to know something very important. There's a big misconception. People are trying to eat fat-free food and all that. But let me tell you that fat is required in small amounts, not large amounts, but small amounts. Um, Fat helps in insulation, cushioning the organs, and giving us energy, all these things. You must read the food labels, and when you read the food labels, they say there are unsaturated fats, saturated fats, trans fats. In unsaturated fats, there are polyunsaturated fats and monounsaturated fats, right? So these are all the categories that you can see on food labels. Out of all these, which ones are good? Unsaturated fats are quite good, and they are found in nuts, fish oil, plant oils, and things like that. Whereas saturated fats and trans fats are bad. Out of these two, trans fats are very harmful to us. So learn the benefits of fats and know that not all fats are bad. Fats are actually required. We need to really consume the unsaturated fats to keep us healthy, because when we eat extra starchy food, our bodies make fats, and in order to eliminate the fats and the cholesterol that liver makes, if it is extra, then in order to remove it, actually we need good fats. Good fats remove bad fats. A fat molecule is made of two parts. One is glycerol, the other one is fatty acid. Remember, guys, fatty acid is different from fat. Fatty acid is part of fat. So what are the two parts or two components of a fat molecule? A fat molecule is made of glycerol and fatty acid. Look at this fatty acid, guys, the yellow highlighted part. Do you see that this is made of hydrocarbons attached to this functional group? Try to recollect this functional group. Yeah, this is carboxyl functional group. Carboxyl functional group is making this entire molecule a fatty acid. That's the name, fatty acid. Okay, when the fatty acid is attached to glycerol by dehydration, they form a fat molecule like this. One, two, or three fatty acids can be attached to glycerol. When three fatty acids are attached to a glycerol, then it becomes triacyl glycerol or triglyceride. So remember, one fat molecule, that is triglyceride, contains one glycerol plus three fatty acid chains. And these are not monomers. Coming to fatty acids, there are two types, saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have all single bonds. Unsaturated fatty acids have one or more double bonds. I'll show the picture in a minute. Before we go there, remember fatty acids are also required. The best examples are omega-3 and omega-6. These are found in fish oils, olive oils, and other good fats. So look at this. This is a unsaturated fatty acid. Because it is connected to glycerol, this entire fat molecule becomes unsaturated fat. Because of this double bond, there is a bend in the molecule. So this entire molecule is bendy. And because molecules with double bond, fat molecules with double bond, they're bendy, they are not able to pack closely together at room temperature. Hence, they remain as liquids at room temperature. So I want you to repeat this part and learn this, and also apply exactly opposite uh, points to saturated fat and learn why saturated fats are solid at room temperature. And I would like to see this in your notes. Answer the question, why unsaturated fats are liquid at room temperature, why saturated fats are solid at room temperature. You have to explain the basis in its structure and the shape of the molecule. Coming to the next subcategory of lipids, which are phospholipids. Phospholipids are the lipids which make cell membrane, organelle membrane, bacterial cell membrane, anything in living organism, whichever is saying membrane, the major component of the membrane is phospholipid. Each phospholipid has a oval shaped head and the tails, and in a membrane always tails face each other and heads face away. The reason why tails always face each other in a membrane is because the tails are hydrophobic because of nonpolar covalent bonds, whereas heads, they are hydrophilic. And they're hydrophilic, so they like water, so they are in contact with water outside and inside, right? And remember, membrane always, whether it is cell membrane, organelle membrane, nuclear membrane, bacterial cell membrane, all of them, they are made of two layers of phospholipids. Coming to the structure of each phospholipid has two fatty acid chains attached to glycerol, which is attached to phosphate, and that phosphate is attached to choline. Yes, you have to know all these labelings for the test. The entire structural formula of phospholipid. You need not know the space filling model, but the short form of phospholipid, oval hydrophilic head and hydrophobic tails together makes a phospholipid. Two layers of phospholipids with their hydrophobic tails facing each other makes a membrane. Remember, cell membrane is liquidy. It's because these phospholipids, they are just staying together because they want to stay away from water. Their tails want to stay away from water. There's nothing that is keeping them together. Third subcategory of lipids is steroids. Steroids do not contain glycerol or fatty acids. Look at this, guys. It contains 4 fused rings and all these methyls and this carbon chain and hydroxyl and all that. Examples of steroids are now cholesterol. When we consume cholesterol, we are able to make estrogen and testosterone. That is the starting ingredient for making our hormones. So cholesterol is required. Just because I said cholesterol is required, don't start consuming a lot of cholesterol, guys. It is not good. Cholesterol, actually, we need not even eat it because our livers synthesize cholesterol. Okay. But children need cholesterol. Children, babies, children, growing children definitely need cholesterol because our cell membrane contains cholesterol. On top of phospholipids, our cell membrane also contains cholesterol. Um, so that's the end of lipids. Yeah, for those who are hearing the uh, online lecture, you are probably not able to participate in this think pair share activity in the classroom, so I want you to answer these questions in your lecture notes and show me your lecture notes in your next visit. Proteins. We will cover this proteins in the next video.